Chapter 1 Molecules of Life, Subtopic 1.4 Proteins. Learning Outcomes. At the end of this lesson, students should be able to describe the basic structure of amino acids, state how amino acids are grouped, and classify proteins according to structure and composition. Protein is unbranched polymer. It made up of many amino acids. These amino acids are joined together to form a long chain known as polypeptide. Protein consists of one or more polypeptide chains which folded and coiled into specific conformation, mostly composed of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Basic structure of amino acids. Amino acids consist of 4 components attached to a central carbon. These components include an amino group, a carboxyl group, a hydrogen atom, and a variable R group or side chain. Carboxyl group give acidic properties to amino acid, while amino group give basic properties to amino acid. Amino acids are amphoteric. It has both acidic carboxyl group and basic amino group. How amino acids are grouped? There are 20 types of amino acids commonly found in proteins. All have the same basic structure but differ in the side chain R. Amino acids are grouped based on the properties of R group.
All proteins are generated from smaller units, known as amino acids. Their name originates from the most important chemical groups, always present in their structure. The amino group, NH2, which is basic, and the carboxyl, COOH, which is acidic. At the center of all amino acids, there is an atom of carbon, alpha carbon, to which the amino, NH2, and carboxyl, COOH groups, an atom of hydrogen, and another group, the so-called side group, frequently denoted as R, are joined. The differences between particular amino acids depend on the structure and properties of the side group.
There are 4 groups of amino acids: nonpolar, polar, acidic, and basic. Nonpolar amino acids are hydrophobic, if not soluble in water. R group or side chain usually consists of hydrogen and carbon atom. Example, glycine. Polar groups are hydrophilic. It can dissolve in water. Side chain usually has OH group. Example, serine. Acidic group has COOH group or carboxyl group in side chain. Example, aspartic acid. Whereas basic group has NH2 group or amino group in side chain. Example, lysine. Diagram shows 20 types of amino acids. All amino acids have same basic structure but different in side chain. Nonpolar, polar, basic, acidic. Formation of the dipeptide. 2 amino acids are joined by condensation process to form a dipeptide. In this process, a hydroxyl group from the carboxyl end of one amino acid and a hydrogen from the amino group of another amino acid are removed. A covalent bond is formed between carbon and nitrogen. It is also known as peptide bond. Dipeptide is hydrolyzed into 2 amino acids by hydrolysis process. meaning water is added in this process. As a result, peptide bond is broken down and produces 2 amino acids. Diagram shows formation and breakdown of the dipeptide. During formation of dipeptide, hydroxyl group form the carboxyl end of one amino acid and a hydrogen from the amino group of another amino acid is removed as water by condensation process. As a result, 2 amino acids are joined together by peptide bond, while water molecule is added to break peptide bond during hydrolysis process.
The peptide bond involves the carboxyl, COOH, and amino, NH2 groups of the 2 amino acids lying next to each other in the peptide chain. When the peptide bond is formed, only rigidly covalently connected CO and NH groups stay in the compound generated. The rigidity of the peptide bond is a very important feature of the three-dimensional secondary structure of proteins. The rigidity causes the oxygen atoms and also the hydrogen atoms of the 2 peptide bonds on each side of one amino acid to be oriented in opposite directions. 2 amino acids create a peptide bond between themselves in a process known as condensation, which involves the amino NH2 group of one amino acid and the carboxyl group of the other one. The products of this process are one molecule of water and a dipeptide. The dipeptide synthesized can react with other amino acids or with other peptides or proteins to form longer molecules. Peptides consist of a small number of amino acids. Longer chains are called polypeptides, while proteins may be formed from one or more polypeptides and are generally functional units. Note that dipeptides and bigger peptides preserve the amino NH2 group at one end and the carboxyl COOH group at the other. In biochemistry, those tails are often called the N-terminal, which is at the amino end of the amino acid, and the C-terminal, which is at the carboxyl end of the amino acid. There is a convention that every peptide is always described from the N to the C-terminal. In living organisms, the process of condensation usually requires large proteins with catalytic activity, known as enzymes. The main chain of a peptide, polypeptide, or protein, which involves alpha carbons, peptide bonds, and atoms of hydrogen, is called a backbone.
Classification of protein. Based on their structures, protein can be classified into 3 groups: fibrous, globular, conjugated. Fibrous protein mostly are secondary structure. It forms long parallel filaments or strands. This protein is insoluble in water, but the structures are stable. Fibrous protein give mechanical and structural support to organisms. Examples of fibrous protein are keratin, actin, myosin, collagen, and silk. Globular proteins mostly tertiary or quaternary structure. It folded to form compact spherical shape. This structure relatively unstable. Globular proteins are relatively soluble, forming colloid in water. It functions in metabolic and chemical processes. Examples of globular protein are enzymes, hormones, antibodies, and hemoglobin. Conjugated proteins refer to proteins that can function well if it joins with other compounds. The compound is non-protein compound, which is known as prostatic group. Examples of conjugated proteins are glycoprotein, lipoprotein, chromoprotein, and nucleoprotein. Glycoprotein are proteins that contain polysaccharide as its prostatic group. It can be found in cell membrane and mucin in saliva. Lipoprotein are proteins that contain lipids. It can be found in cell membrane and act as lipid transporter in blood plasma. Chromoproteins are proteins that contain pigments such as myoglobin and hemoglobin, while nucleoproteins contain nucleic acid such as chromosome and ribosome.